Hello, everyone. Whoa. Sorry, that was loud. Thank you all for coming today. It's great to see so many people here. Our speaker today is Dr. Sean Connolly, who is joining us from the Smithsonian Tropical Research Institute in Panama, where he is a senior scientist. Dr. Connolly earned his PhD at Stanford with no other than our own Nate Sanders. And since then, he's done a lot of really great research into coral reef ecosystems and the mechanisms, the maintenance of biodiversity in coral reef ecosystems, and the impacts of human activities. Today, he will tell us more about his research in his talk, Commonness, Rarity, and Biodiversity on Indo-Pacific Coral Reefs, Confronting Ecological Theory with Data and Species-Rich Ecosystems, or Systems, sorry. If you would all like to welcome me, sorry, I'm nervous. If you would all like to join me in welcoming Dr. Sean Connolly.
Okay, thanks, Sean. Can everybody hear me? Yep, okay, great. So ecologists, as you all know, have long sought to explain patterns in the commonness and rarity of species. Why are some species common? Why are other species rare? Why are some pairs of species common together and rare together, and other species are not? And so to attempt to explain those things, theoretical ecologists have tried to construct models to understand the dynamics of species. And classically, they would adopt an approach something like this, right? They would write down an equation to describe the dynamics of each one of the populations in their community, and they would express those dynamics as functions of things like species-specific carrying capacities, the rate at which they recover from excursions to very low density, and interaction terms that represent the effect of an individual one species on another species' capacity to contribute to population growth. Limitation of this approach is that it won't work in highly species-rich systems. And one reason it won't work is because of something called the curse of dimensionality. So if I have a community and that community consists of only two species, I only need to estimate the strength of two different linkages, the effect of individuals of the green species on the orange species, and vice versa. On the other hand, if I have a community of 4 species, I now have 12 different linkages that I need to estimate. 6 species, I have 30 linkages. And if I have a community of 100 species, which is not that uncommon in some tropical ecosystems, such as the rainforests and coral reefs in the background there, I would need to estimate around 10,000 linkage strengths. You can see the number of quantities I need to estimate grows much faster than the number of observations that I have as the number of species increases. Additionally, many of these species are going to be very rare, further complicating the problem of estimating interaction strengths. So for that reason, macroecologists, in seeking to understand biodiversity, have adopted a somewhat different approach, and that is to make assumptions about the overall distribution of demographic characteristics, interactions, and so forth in a community, and then derive from those assumptions aggregate properties of communities. And so what I mean by this is, so here's an example. This graph here is called a species abundance distribution. It's just like the number of species represented by different numbers of individuals in a sample. So rather than try to explain which species should have which abundance in a community, we simply say, well, what proportion of the species in the community are going to be really rare? What proportion are going to be common? How common will the most abundant species be? And so forth. Now, the most aggressively simplifying, I guess, of these sort of broad-scale theories of biodiversity is neutral theory of biodiversity, developed by Steve Hubble and Graham Bell and their collaborators in the mainly in the sort of late 1990s. And the basic idea here, the basic idea behind neutral theory, is that one simply assumes that every individual in a community, so here's a hypothetical community, each color represents a different species, each little fish is an individual. So all individuals in this community are assumed to have the same probability of producing 0, 1, 2, 3, 10, 50 offspring as every other individual. Any individual in the community has the same chance of dying between this year and next year as any other individual. And any place an individual of one species can live, an individual of any of the other species can also make a living in that spot as well. And so given those assumptions, any differences among species that I see in abundances have to be due entirely to demographic stochasticity, that is, chance variation in the fates of individuals. So which individual's offspring happened to be closest to a site when it became available for colonization, or which individual happened to be in the path of a hungry predator at the wrong time, and so forth. Whoops. So early in my career at James Cook University in Australia, where I first started working on coral reefs, we thought we would use coral reef ecosystems as a framework for testing neutral theory, and we published this paper, which says coral reef diversity refutes the neutral theory of biodiversity. Essentially, we argued that neutral theory could not simultaneously explain variation in abundances among species and spatial beta diversity turnover patterns without making inconsistent assumptions at those scales. So we thought, okay, great, on to the next thing. But then Igor Volkov and various collaborators, including Steve Hubble, published a paper where they said, well, hey, we have a different version of neutral theory, slightly different model, and it actually explains those patterns after all. Now, with respect to this paper, I think they only managed to do this by making an assumption at one spatial scale that is inconsistent with the results of their model fits at a different scale. But more generally, a lot of this happened early in the 2000s. Someone would write a paper saying, well, I tested neutral theory on my system and it totally fell over. And then someone else would say, aha, but I have a different neutral model and it actually explains your data after all. So I think we can understand this by making an analogy with the theory of scientific research programs in philosophy of science made popular by the philosopher Imre Lakatos. And so the basic idea here is that any theory consists of a hard core of assumptions that essentially define the theory, right? So in order for a model to be a neutral model, it needs to have two things. It needs to assume that every individual is demographically equivalent to every other individual. Everybody's got the same chance of giving birth and dying and so on. And ecological equivalence, that is, any place an individual of one species can live, an individual of any other species can also live. Without those assumptions, you might have a model, but it won't be a neutral model. But in order to produce a model that can predict patterns in abundance, those core assumptions have to be packaged with what we might call auxiliary assumptions. So for example, how do new species enter the system? How are individuals arranged in the landscape or seascape? How do they disperse over that space? And whether and how individuals compete with each other for resources. And because different neutral models can make different auxiliary assumptions, they can potentially make different predictions about abundance patterns. Right, so I did not want to spend the rest of my career playing whack-a-mole with different versions of the neutral theory of biodiversity. So I started trying to figure out if there was a way to identify shared predictions of models that made those core assumptions of neutral models. And it turns out that there was a paper published in Oikos in 2006 by Salvador Pueyo, and in that paper, Pueyo proposed that models, based on some mathematical arguments, essentially involve taking a model of pure demographic drift and then adding successive, essentially higher order expansion terms around that model. I'm not going to go into the details of it, but suffice to say, his main thrust was the distribution of relative abundances in a community. If you plot a frequency distribution of species abundances, you should get something that looks like a gamma distribution if that community follows neutral dynamics. Conversely, as communities become increasingly non-neutrally structured, that frequency distribution of abundances should start to look progressively less like a gamma distribution and more like a log normal distribution. Well, I mean, so that sounded great, but it was based on a mathematical approximation, and so I was not fully convinced that this was right. So what I did is I went to the literature. I grabbed a bunch of different neutral models. Those are the different colors here represent models that all make the core assumptions of neutral theory, but that differ in those auxiliary assumptions that I mentioned. And so I simulated data from those different neutral models, and then I said, well, does a gamma distribution actually characterize the frequency distribution of abundances better than a log normal distribution in accordance with what I'm calling Pueyo's conjecture? I'm going to plot the relative support of the gamma distribution versus the log normal. So bigger numbers mean, I can't remember the order, so I'll have to tell you when the points come up. And I'm going to plot that as a function of the median number of distinct abundance values in the community. So what do I mean by that? Well, if I have a community with 10 species, 9 of them all have abundance 1, and 1 of them has abundance 67, I have a frequency distribution with 2 bars. And so I'm not going to be able to distinguish between 2 alternative distributions as characterizing that community. And so here's the graph. So basically, this is relative support of the log normal compared to the gamma. So 0.5, the dashed line, means the 2 models fit the data equally well. As you move down, you become increasingly confident that the gamma distribution characterizes a community better than the log normal. So there's a bunch of points for each color. Those are simply different combinations of neutral model parameters that were designed to span the range of community sizes and species richness values that you would typically see in species abundance values in the literature. What you can see is that if you start out with, when I have very few distinct abundance values, I really can't tell what's going on. But as I can increasingly resolve the shape of the frequency distribution of abundances, because I have more distinct abundance values in my histogram, I become increasingly confident that that distribution is better approximated by a gamma distribution than a log normal distribution. Okay, great. So what about empirical data? So I wanted to then compare that with real species abundance data to see if they looked like the neutral data, right? The data simulated in accordance with a bunch of different neutral models. And so I did that using data from the Census of Marine Life project. So this has got a bunch of data contributors. There are 14 different marine data sets here, ranging from intertidal to abyssal depths of the sea, a whole bunch of different taxa, thousands of species, and so on. So here's what happened when I used data simulated from neutral models, so data that I know accord with the assumptions of neutral theory. And then this is what happened when I did the same thing with the real data from marine communities. So the different color symbol combinations represent different data sets, and then the different size symbols represent those data aggregated at different scales because the sampling designs were often hierarchical. And so what you can see is what's going on the right is that the more I can resolve the shape of the abundance distribution, the more confident I become that the distribution is much better described by a log normal than by a gamma distribution. Very much different from what I saw with the neutral model simulations. So what does all that tell us? Well, what indicates to us that neutral theory by itself is not sufficient to characterize patterns of commonness and rarity in the marine biosphere. Another way of saying that is that trait-environment interactions must be playing some kind of a role, right? So we can't simply assume species are all identical and explain abundance variation. What it doesn't tell us is what the relative importance of those things are. So maybe mostly chance fates of individuals, mostly demographic stochasticity driving what's going on, but there's actually ecology that's important at the margins, or maybe trait by environment interactions are what's actually determining relative abundances, and it's the neutral processes, the demographic stochasticity, that's creating a little bit of noise around that. So to answer this question, we need to move beyond static abundance distributions, and in particular, I want to make the argument that the temporal dynamics of relative abundance, how relative abundances change through time, contains information about the importance of species differences to relative abundance patterns. So this is a hypothetical community. Each line is meant to represent a different species. The abundances, some measure of abundance is on the vertical axis. Time is on the x-axis. So you have the abundances of these different species varying through time. So this is a community that is where you could say the relative abundances are highly deterministically structured. There's something about the red dotted line species that is making it consistently abundant in the community, and the dotted green line towards the bottom, something about it is making it be persistently rare. So if I took a snapshot of this community at time 0 and time 1, and I asked how similar are the relative abundance patterns, I would get a large number. I would get a correlation of abundances close to those two communities are very similar to each other. If I took another snapshot at time 10 or time 20 or time 30, the community would still look very much like how it looked at time 0. So community similarity, right, through time is going to tend to be very, very high, regardless of how long I wait in between snapshots of a community. Here's another hypothetical community. In this community, we also have, I think, 10 species. In this case, and the variance in abundances is the same in these two communities. But in the second community, the average abundance of every species is the same, and the variance is produced because they're all bouncing up and down in different ways in response to fluctuating environmental conditions. So if I did the same exercise here, I take a snapshot of the community at time 0, another snapshot at time 1, another snapshot at time 10, 20, 30, what I'm going to find is that, well, initially there's going to be some ecological memory. There's some temporal autocorrelation in abundances. So the community at time 0 and time 1 might be pretty similar to itself. But the longer I wait, the more those abundance fluctuations shuffle around the relative abundances, and the community similarity is going to decline toward 0. So that is the basic intuition. So this intuition was leveraged by Steiner-Engen and Engen's various collaborators, largely working at the Center for Biodiversity Dynamics in Trondheim, who worked with a particular stochastic community dynamics model. It looks like this. Don't let the formalism freak you out too much. Basically, is just characterizing the change in abundance of some species in the community, where the abundance is expressed on a log scale as a function of that population's capacity to grow from low density values. There's some density dependence operating that stops the population from growing forever. And then there's this extra term on the end, this sigma E DWE, which represents the effect of environmental fluctuations on the, if you like, the intrinsic growth parameters. So that sigma E tells you how sensitive the species is to fluctuations, how sensitive the species' intrinsic growth parameter is to fluctuations in the environment. So how much does little r bounce up and down over time? And so an advantage of this framework is it allows for some species differences without having to specify species demographic rates on a species by species basis, which is that whole cursive dimensionality thing that we're trying to avoid. It does that by basically putting distribution on a model parameter. So like, we don't know which species has which little r, but we know that in this community, little r varies according to a normal distribution with some mean and some variance. Another convenient thing about this model is that it does tend to produce, well, it does produce a log normal distribution of species abundances, which is consistent with what we saw at least in the marine species abundance data that I showed you a while ago. And then finally, Engen and colleagues showed that one can use a community time series, a time series of relative abundances, to partition the variance in species abundances by leveraging the intuition that I showed you on the previous slide. So, whoops. More specifically, the variance in log abundance in the community is a sum of 3 different terms. That first term, which I would call sigma squared deterministic, is basically the variance in abundance that is due to variance in the long-term average abundances of the different species. So if you like, this is like the among species variance in their carrying capacities. The second term, what I'm calling sigma squared ENV, is basically the variance that is due to environmental fluctuation. I'm going to call that response diversity, so it's the variance that comes from the fact that the different species are bouncing up and down in different ways in response to environmental fluctuations. And then there's a third term, which you could think of as sort of over dispersion. This represents, can represent effects of, say, non-random sampling, right? Tendency, if you take a sample over here and then take another sample over here, you're going to get some additional variation that is not due to purely random sampling because of aggregation effects and stuff like that. Now, neutral processes are essentially going to partially influence these second 2 terms, the blue term and the green term. The green term, because neutral variation produced by demographic stochasticity scales with population sizes the same way as over dispersion, but also will produce some temporal autocorrelation. So in that sense, it's a little bit more like the sigma squared ENV term. And so it turns out we can estimate these quantities by examining how a community's similarity with itself changes with the time lag in between my snapshots of that community. Specifically, if I fit a model where the community similarity decays over time towards an asymptote, that asymptotic value is that deterministic component, the variance in abundances that is due to persistent long-term variation in mean abundances due to persistent demographic differences between species. The difference between the intercept and the asymptote represents this environmental effect, so mainly response diversity with a little bit of impact of neutral or demographic stochasticity. And then the remaining term is this over dispersion contribution. And so what that means is if we have a snapshot of community through time and we can fit this sort of a model, we can estimate the relative importance of these different types of processes in determining commonness and rarity in the community of interest. So a PhD student who was working in my lab at the time, Cheng Han, is now at National Cheng Kung University in Taiwan, where he's an assistant professor. So he was interested in using this approach to try to characterize the structuring of reef fish communities on the Great Barrier Reef. And to do that, we worked with data from the Australian Institute of Marine Science long-term monitoring program, where they surveyed 40 different reefs up and down the Great Barrier Reef. The data I'm going to show you from this paper focuses only on the 11 years where sampling was annual, but we've since expanded the analysis to look at the full time series, including biannual sampling, which spans, I think, a total of about 30 years or so. So this graph is a frequency distribution among the reefs. So what we did is for each reef's time series, we estimated the variance components, and then we plotted the frequency distribution of those variance components, so the number of reefs. So each reef contributes a value to the red histogram, a value to the blue histogram, a value to the green histogram. And so what you can see from this is that persistent species differences, that is actual biological heterogeneity among species, is overwhelmingly important in structure. It explains about 3/4, on average, about 3/4 of the variance in log abundances in the communities. And then only about 20% is due to this response diversity term, and only about 7% to this over-dispersion term. And since neutral processes can only enter by partially influencing those last two terms, we can conclude that neutral processes account for a much smaller proportion of the total variance in species differences. Okay, sorry. So I should say there is still a lot of variability among reefs in these variance components. So Cheng-Han also wanted to know why that might be, and it turns out that the extent to which a reef fish assemblage is deterministically structured depends upon how volatile coral cover was over time on that reef. So in this graph, again, each reef contributes a red circle, a blue triangle, and a green X, representing the variance components. And you can see as the temporal volatility of coral cover increases, the sort of, if you like, the niche structure of the assemblage is eroded, or the importance of response diversity increases. Conversely, if we look at, somehow a timer got started there. But conversely, if we look at standard measures of alpha diversity, like species richness or evenness, there's no relationship with volatility in coral cover. And we also don't see a species composition varying depending on coral cover volatility. These quantities respond much more to standard macroecological gradients like proximity to coastal influences or latitude. So this tells us that firstly, that it's possible to accommodate species differences in a tractable stochastic theory of community dynamics can serve as an alternative to neutral theory. When we apply this theory to reef fish assemblages, we do see this dominance, this overriding importance of demographic heterogeneity among species as determinants of commonness and rarity. And we also found that the assemblage structure of fishes did seem to be destabilized by volatility in coral cover, and that these variance components of community structure are responding to different environmental drivers, that is, variation in community structure components among reefs is driven by different environmental drivers than variation in species richness or evenness or species composition, suggesting that they may provide a sort of complementary early warning sign of adverse ecological change. Okay, so we're not done yet, of course, because we got all this by applying Engen and colleagues' stochastic dynamic theory, which I've shown you here. But like any theoretical model, it makes some assumptions, and so we need to worry about whether violation of those assumptions in our system might be leading us to draw erroneous conclusions. So one of those assumptions is that species interactions are negligible, and that is, if you look at the equation up here, the dynamics of abundance of species I depends on the abundance of species I, but not on the abundance of any of the other species in the community. So essentially assuming, well, we can ignore the effect of interactions between species in determining abundance patterns. A second one is that species are responding independently to environmental fluctuations. That is, in this model, there are no positive or negative correlations on how species bounce up and down in response to the environment. And a third point is that all of the heterogeneity among species in this model is driven by variation among species in this intrinsic growth parameter, like how fast populations recover from low density. But there are two other important parameters in the model. One is this delta, and the other is sigma. And so the model is assuming that the strength of density regulation and this sigma E, which is basically how much does your little r bounce up and down because of environmental fluctuations, that those are the same for all the species in the community. Whereas we know that some species tend to be more volatile, they tend to bounce around more in response to environmental fluctuations, and others less so. So, one question we might ask is, well, how strongly do our communities actually violate the assumptions of this non-neutral stochastic dynamic theory? And then accordingly, how robust are our estimated variance components to those assumptions? And to what extent are long-term abundances driven by either differences in those intrinsic population growth parameters? I've called them rates. I think technically they're not rates here, but differences in the strength of density dependence, versus differences in the strength of, remember we assume in Engen's model, all the variation among species is driven by that first bullet point, differences in intrinsic growth parameters, nothing in variation in density dependence. And also when we consider relative abundances, to what extent are the dynamics driven by species interactions, which we've assumed are negligible, versus response diversity, which we've assumed is extremely high. We've essentially assumed they're all fluctuating independently. Okay, so I'm now going to talk about the work that was led by a different PhD student, Alfonso Ruiz, who just finished his thesis and will be starting a postdoc in Helsinki next month. So what Alfonso wanted to do was see whether he could relax the simplifying assumptions made by Steiner, Engen, and colleagues without sort of collapsing back into the problem of the cursive dimensionality. And so Alfonso worked with a model that is structurally very similar to the model that Engen and colleagues worked with. So so technically we'd call it a Gompertz, multivariate Gompertz model. This model is written in discrete time, so it's going to look a little bit different. But basically, you have a vector of species abundances at time t. Abundance of species, log abundance of species 1, species 2, species 3, and so on. Then we have a vector of the species abundances at previous time step. And then we have a vector of those intrinsic growth parameters, like how fast do the populations recover from low density. So again, this is a vector. This was little r in the other model. Then we have the species bouncing up and down in response to environmental fluctuations. And lastly, we have this B term, which measures the strength of interactions, the strength of biotic interactions in the system. And previously, we assumed that these interactions were only between individuals of the same species. We want to relax that assumption. So this would be a sort of full model. You have each one of those subscripts would represent a species. So a 3-species community would look like this. The diagonal elements would say, what's the effect of species 1 on itself? That is the within species density dependence, species 2, species 3. We assumed that these 3 values were the same in the previous model. So we want to allow these values to vary between species. We also assumed that the between species interactions were negligible. So we assumed these were all 0. We want to not have to force all these to be 0 without having to estimate each one of them individually, which is the cursive dimensionality problem. Okay, so for the responses to environmental fluctuations, we have in any given year, there's a positive or negative perturbations in the population growth of the different species. So we'll model those perturbations as coming from a multivariate normal distribution. So again, the diagonal represents the variance, like how much does little r bounce up and down for each of the species. We don't want to have to assume that that amount of bouncing up and down is the same for everybody. We want to allow some species to maybe have higher variance and some to have lower variance. And then these off-diagonal elements are the covariances. And so the previous model assumes all the fluctuations are independent. We want to allow these to not necessarily all be 0, again, without having to estimate them all one by one. And so these two matrices, because they're square matrices, the number of parameters I need to fill up the matrix grows with the quadratic of the number of species. That's the whole cursive dimensionality problem that we want to avoid. So we need to somehow reduce the dimensionality of the B and sigma matrices. So to reduce the dimensionality of the species interaction matrix, Alfonso used something called a regularized horseshoe prior. So if you're not used to dealing with Bayesian models, you can think of this as like a random effects distribution. The horseshoe prior has a particular functional form, which is to say it has a really sharp peak at 0 and then thick tails. And the basic idea is that in a high diversity community, most species are probably interacting pretty weakly, but some species might have strong interactions. So the structure of that horseshoe prior is that for species interactions where there's not much evidence of interaction, the shape of the horseshoe prior will shrink those estimates toward 0, toward where the peak in the density is. But where there is evidence of interactions, then those particular pairwise parameters can escape that shrinkage to 0 and have meaningfully positive or negative values. Okay, for the response diversity, we didn't want to make the assumption like we did for species interactions that, well, most of these are probably close to 0. So the approach that Alfonso took was a little bit different. So here's the variance covariance matrix. The diagonal is the variances, how much do individual species bounce up and down. The off diagonal is like how positively or negatively correlated are those variances. So the basic logic here is that this matrix of variances and covariances arises because you have a bunch of species whose population growth rates are being pushed around by some environmental drivers that are unmeasured. So we haven't measured these environmental variables, but presumably its environmental conditions are changing, and that's causing the demographic rates to bounce up and down. And that's creating the correlation structure. So we're going to treat those unobserved drivers as latent variables, and then we're going to construct a matrix. So here's an example where there are 3 latent variables. So we now have the number of species and the number of latent variables. What we need instead of the full matrix is we need to know how sensitive is each species. What is the coefficient that describes how the species population growth rate depends on the value of each one of the 3 latent variables. And then we'll also have to estimate the latent variable values. But filling up that matrix on the right, even if we also have to estimate latent variable values, is still a lot less parameters than filling up the matrix on the left. And so this maybe sounds a bit like voodoo, because I'm saying like we have this big variance covariance matrix driving by these things that we can't observe, but we're going to estimate the things that we can't observe, and we're going to figure out the coefficients and everything else. But in truth, it's not that different than what any community ecologist does when they do a PCA, right? So if you're a community ecologist, you go out to like say 5 different sites, you measure the abundances of a bunch of different species, right? You have 20 species, you have a 20-dimensional data set. What do? You feed it to a PCA. The PCA then gives you a plot where each one of your 5 communities has got a PC1 score and a PC2 score, right? You didn't go out and measure PC1 in the field or PC2 in the field. What's happened is the model is positing the existence of these latent variables and using the higher dimensional information in the species abundance data to try to infer PC1 score, PC2 score. So that is basically the role. So essentially, the latent variables, the latent environmental variables, are working much like the principal component axes in a PCA. That's essentially how it works. So Alfonso was able to get this model to converge for the 40 most prevalent species in the data, which accounts for about 80% of the total number of individuals. And so for reef fishes on the GBR, one of the key things he found was that the between species interactions are overwhelmingly very, very close to 0, especially relative to the strength of between species interactions, which were consistently compensatory interactions, as they indicate that individuals have negative effects on other individuals' population growth rates when they belong to the same species. So you have strong intraspecific, detectable intraspecific density dependence, essentially negligible species interactions. Secondly, response diversity is actually pretty high. So the correlations in how species respond to environmental fluctuations are not zero, not perfectly independent, but you'll see they're mostly between 0 and 0.5. So they're relatively low. They're not all close to 1, which would be a community with very low response diversity. We also found that species do vary in their intrinsic growth rates, as assumed in Engen and colleagues' model, but they also vary in their within-species density dependence and in their sensitivity to environmental fluctuations. So to get a sense of the comparative magnitude of that variation, this is the density-independent growth parameter. Coefficient of variation is about 0.5. The density-dependent component of population growth, the strength of density regulation, coefficient of variation, pretty similar, about 0.6. And the coefficient of variation of that environmental standard deviation parameter, how much population growth rates bounce up and down in response to environmental fluctuations, coefficient of variation about 0.6. So they are, all three of these parameters vary among species to similar degrees. Now, when I first saw these results, I was incredulous that a model would be able to successfully distinguish between the effects of species interactions from the effects of environmental fluctuations. So I made Alfonso do a bunch of simulations. And so in one set of those simulations, he said, okay, let's have a community where all the environmental correlations are 0. The species are responding perfectly independently to environmental fluctuations, and 10% of the interactions are going to be non-zero. 90% will be 0, 10% will be non-zero. And so what we want to know is if we simulate data like this and then we fit our model to it, does the model correctly identify the non-zero species interactions and correctly realize the environmental correlations are 0, or will it detect the covariation produced by the species interactions and put that in the environmental covariance matrix incorrectly? And so these are the results of his simulation. So the environmental correlations, which in the model are 0, so the true value is the red line, are 0. And you can see that in the pairwise correlation estimates, none of them are significantly different from 0. They're noisy, but none of them differ from 0. All the confidence intervals overlap 0. And similarly for species interactions, the 90% that were 0 are estimated to be very, very close to 0. The ones that were, these are the true values used to simulate the data. These are the estimates that were produced when the simulated data were fitted to the model. And so what you can see is the ones that were positive were estimated to be positive. The ones that were negative were estimated to be negative. The ones that were estimated to be really positive were really positive, and so on and so forth. So it does a remarkably good job of correctly figuring out what's driving the structure in the relative abundance dynamics. He then turned that around and said, well, what if I set all the species interactions to 0 and I generate non-zero environmental correlations randomly? And so that's what's shown here. So all the species interactions were estimated to be 0, and the estimated correlations were in fact quite well correlated with the true environmental correlations used in the simulation. Little bit noisier than the species interactions case, but for the most part, it's doing a pretty good job of telling us what is structuring the fluctuations in relative abundances. So it seems to be working pretty well. So this tells us, what does it tell us? Well, basically, what I take away from this is that coral reef fishes are exhibiting classically Gleasonian dynamics. So I don't know whether you learn about Gleasonian versus Clementsian or Eltonian community structures, but the basic idea here is that interspecific in the Eltonian or Clementsian view, communities are essentially tightly interlinked assemblages. In the Gleasonian view, species are essentially responding individualistically along environmental gradients. So in this case, interspecific interactions are very close to 0, and they're much smaller than the within-species effects. And there's pretty strong response diversity. That is, species responses to varying environmental conditions are relatively weakly positively correlated. So they're pretty different from each other. And a second point is that density-dependent processes do seem to be important determinants of patterns of commonness and rarity. So there's a meaningful variation in the strength of the density-dependent parameter that is contributing to variation in species relative abundances. So when I was a PhD student, there was a lot of interest in sort of trying to come up with a unified theory of ecology. And so you would read these little news articles in Science or whatever about like the group of theoretical ecologists who wanted to come up with the fundamental theory of ecology, which would be like 5 equations that would explain everything about abundance dynamics. And so they would always interview Simon Levin to get that point of view. And then there was this other view that said that, no, it's more like an Earth system and we're going to have an atlas of models. And then they would talk to my old supervisor, John Roughgarden, about that. And so we're not arriving at a unified theory of biodiversity. We're still estimating parameters from data, but nevertheless, like I'm surprised that we're in a situation where we can actually get estimates. Like we're getting out of this process estimates of all the different species interactions, even though we don't have to estimate them all separately, even though we're using dimension reduction techniques that allow us to extract this information without being tripped up by the curse of dimensionality. Secondly, the second point I want to make is that we can extend this approach to look at trait by environment effects on those parameters, which is something we like if environmental conditions are changing. So if we want to understand how ecological communities are going to respond in the future, we need some way of connecting environmental conditions to demographic rates of species so that we can ask the question, how are these communities going to look different as environmental conditions change? And so what I presented you, the environmental variables were latent variables. We didn't know what they were, but if we have candidate environmental variables, we can plug those in as potential explanatory variables for either spatial variation or temporal fluctuations in environmental conditions. In this case, this is a model where species have been classified into different trophic groups, and we're estimating how their intrinsic growth parameters vary as a function of, say, fluctuations in abundance of macroalgae or coralline algae or hard coral or turf or whatever. So for example, if you are a corallivore, you eat corals for a living, then if hard coral cover increases, that's good for your population growth, right? So we can begin to get a handle on how species combination of traits influence not just abundance, but the underlying demographic rates that will determine how those populations will respond to fluctuating environmental conditions over time. So with that, I want to acknowledge a whole bunch of people for work on coral reef community structure, reef coral community structure, all the data contributors and people who wrote code for me for the Census of Marine Life, and my collaborators on the reef fish community dynamics work, including three former PhD students, and definitely the Ames long-term monitoring team and my collaborators at Ames. So thanks, everybody. That's
Oh, there are 2 buttons. Oh. IT is on the way. I think I got it. Yep. Hello?
Yeah, got it.
Sweet.
Thank you. We're doing Lord of the Flies. We're going to pass that around if people have questions.
Cool. Thank you. That was amazing. Are there any questions for Dr. Connolly?
So you have a lot of evidence overall for strong density-dependent interactions within regulating individual species, but not a lot of evidence for them interacting. So you've got like 2 views here, and maybe some others, but you got one view where species are just kind of doing their own thing, nonetheless with strong density dependence. But another view where they're doing their own thing because of historical interactions, either filtering at the level of the regional community because of the potential for interactions, or evolution driving them into areas of space such that you're not seeing those kinds of interactions today. So how would you, if thinking about what you have in terms of data, how would you get towards being able to test that hypothesis for why species do what they do to the next level of beyond the fact that they are locally showing lots of density dependence within individual species trajectories, but appearing to not interact?
Right. So, I guess the two different ways you could look at that. One is the ghost of competition past argument, right, from Joe Connell. That I don't think you could necessarily use these data to draw a conclusion about that unless you I guess you would look for. Yeah, no, I'm not sure how you would use these data for that kind of thing. If you're talking about filtering, filtering out of species that would otherwise interact, then I think you could look for if you could identify trait combinations that were associated with similar responses to environmental fluctuations, you could actually ask whether there is some kind of overdispersion, not necessarily phylogenetic overdispersion in community structure, but overdispersion in trait values. I don't think you would see that in coral reefs. I don't think there's enough of that kind of turnover up and down the Great Barrier Reef. But that's I think how I would approach it.
Sean, great talk. Do you think there are data that empirical ecologists need to be collecting?
Do I think what?
There are data that empirical ecologists need to be collecting to address some of these overarching questions about the structure and dynamics of communities? And if so, what are those data that we have yet to collect?
Right. So, long-term time series data at the community level are super useful. So, trying to count the abundances of everything and not just the handful of abundant species that you're interested in would be really useful. Obviously, terrestrial systems are blessed with way more time series because marine biologists haven't been working on scuba for that long, and it's hard to get people who are trained who can go underwater and count stuff. So we're in a situation where we're, I think, a lot more data poor than terrestrial ecologists are. So that's one thing that I think would be useful. Another thing that, to me, where you can get information on demographic rates, that saves you the trouble of having to wait for a ginormously long time series that you can try to infer parameters from. So if you can actually measure, if you know growth and survival and fecundity, if you have that through time, then you can connect the actual demographic responses to environmental conditions. I think that's going to be, one, that's going to be a cleaner signal. And secondly, if you're interested in things like species coexistence, where the precise nature of the functional relationships and what life stages are happening at, you can get a handle on all that if you have demographic information. Whereas if you just have, total counts like we have here, so we can't ask questions about coexistence mechanisms with this kind of time series data. Because if we're looking for temporal storage effects, we would be looking at one life stage versus something else on another life stage. So I guess those would be time series are great, but time series of demographic rates are at the per capita sort of, or per colony level, or whatever is appropriate for your taxon, I think would make it a lot easier to identify these patterns of variation in responses to the environment, and then allow us to better anticipate how communities will respond to changes in those conditions.
So since I'm department chair, do you think I should impose a requirement that all PhD students spend 15 years collecting data before they get their PhD?
I think your graduate program would probably get small in a hurry if you did that.
Fair enough, fair enough. Other questions? Yeah.
Well, please join me in thanking Dr. Sean Connolly. Connolly. Oh my
Connolly,
gosh,
yeah.
sorry.
That's all right.
Sorry, public speaking is not my forte.
That's quite all right.